So now we talk about the direct observation of soil bacteria. Different techniques have been used for direct observation of soil bacteria. As we say that only 1% of bacteria are culturable, it means that 99% are not culturable, but we can see them with other methods. What are those methods? Those methods include the light microscopes, fluorescence, or electron microscopes. And these methods revealed that around 100 to 1,000 times higher number of bacteria are visible and are present in the soil as compared to the ones that are viable. Viable means they when you culture, they appear on as colonies. And vast numbers of bacteria are extremely small, although they are structurally intact. So such bacteria are so small that their diameter is less than 0.3 microns. Structurally intact means they are viable. I mean, they are living. They are alive. However, because all their structures are okay for starting a life, however, they don't grow. And these are very small cells. And we would call them as dwarf cells in this lecture onward. The methodology in direct observation are different, as you see, and they have different criteria based on which they identify the cell. For example, scanning electron microscopy, SEM, that identifies a cell based on surface and morphology. How is the morphology and how the surface of the cell. And then transmission electron microscopy, that identifies the cell based on the cell membrane or cell wall characteristics. I mean, cell wall or membranes characteristics. Then fluorescence microscopy, that identifies a cell based on a fluorochrome that binds to a certain DNA. So we can see which kind of sequence is present there, and we identify a particular cell. In the same way, confocal laser scanning microscopy and image analysis are very important. And we see that majority of the cells that are visible in direct observations are small. They are dwarf cells, and only 5% of bacteria are larger than 1 cubic micrometer in volume. So regarding the dwarf cells, the cells, the different properties they have include first of all, they're very small. The diameter is less than 3 micrometer. Secondly, they are highly abundant in the soil. As you see, more than 95% are the cells which are dwarf. And then they are highly abundant in the soil, and the abundance increases with depth. And then whenever they are tried for growth, they either don't show any growth, or if they show the growth is extremely slow. Their life cycle is extremely slow, so slow that you can say they don't appear on your plates. It seems that these are the cells that have been exposed to drought for long intervals. So we are not, although we are not sure about that, but there are different hypotheses about it. So secondly, they are stressed by lack of, they are probably stressed by the lack of nutrients. As you know, soil is a very difficult medium to grow, and it's sure that the communities that are present in the soil, they would be exposed, they will be exposed sooner or later to some kind of stress. The major stress comes when there is a shortage of moisture or there's shortage of nutrients. And nutrients, you know, is something which is limited, and same is true for moisture. So so in one of the hypotheses is that this stress because of lack of moisture or nutrient take them to a point where from where they cannot return back to the normal growth. So the frequency of dwarfs and the nutrients are very much related. I can give you two scenarios as an example. First is the nutrient availability and its relationship with the abundance of small cells. For example, when the depth of soil increase, if we increase the soil depth, we see there is a decrease in availability of nutrients. At the same time, the number of viable cells also decrease, and the number of dwarf cells increase. So we see that with decrease in nutrients, the number of dwarf cells increase. Second scenario is the plant growth. When the plant growth is proceeding towards the end of the season, the nutrient availability decrease. And with the decrease in nutrient availability, it has been observed that the number of dwarf cells increase. So there is a very clear relation between nutrient availability and abundance and presence of dwarf cells. While talking about the dwarf cells, if we conclude all our discussion up till now, is that there is a very, I would say a debate among scientists about the nature of dwarf cells. What are actually dwarf cells? Are they the miniature cells of the otherwise normal cells? Some scientists think that they are actually the stressed cells of the same normal cells. When the normal cells are exposed to nutrient deficiency, as we have already discussed, the size of cells decrease due to asymmetric cell divisions. Others think that they are different from normal cells. They are by themselves. They are not the same as the normal cells are. They have their own physiology. These cells exist by themselves. So, according to this theory, they think that these cells are obligate oligotrophs. Oligotrophs are those populations which prefer low nutrient media, which can only grow in low nutrient medium. like the stress soil. So they can grow only in low nutrient conditions. And they have their own DNA intact. And this is what we have already seen, that when we analyze soil, we see a huge number of communities with their sequences which are uncultured. So because they have their own DNA, because they have their own I mean, they are different from the normal ones. So they have their own physiology and they have their own morphology. So this is what this second hypothesis. So if I briefly explain you the dwarf cells here, dwarf cells are the cells which are small in size, and they are the ones which actually don't appear on plates. They are either very slow in growth, or they cannot be cultured simply. You cannot revive their growth. There are different theories about these dwarf cells. One theory is that they are miniature cells of the normal cells that we see on plates. So because of stress conditions, either because of nutrient or moisture stress, their size shrink and they become small. This is one set of theory. The second one is that these cells are different from the normal cells. They are basically normal obligate oligotrophs. They prefer conditions which are low in nutrients, and they have their DNA intact. They have their own physiology and morphology, and they have high surface to volume ratios. What does it mean, surface to volume ratio? It means because their size is small, they have bigger surface to the exposed to the environment, so that they can take advantage of the bigger amount of possible absorptions from the outer environment. So here I explain you a little bit about a method that can be used, or that has been used, to separate bacteria in the soil based on their size. This method was based on the blending and separation of cells from the soil colloids, based on centrifugation and then separation, in which supernatrants are picked, and then again density gradient concentration, which separate the things based on their densities, followed by filtration with different pore size. Different pore sizes allow to separate the different size cells. So now we have discussed about the dwarfs and the culturables. We have already compared, but what are the contribution towards the overall soil turnover in terms of biomass, or in terms of their physiology or metabolism? So this is what I would like to explain here. If we see here in soil, the number of dwarfs is huge as compared to the culturable. The volume of dwarfs, however, is only 10% of the total biovolume. So this is very important. So although more than 95% are dwarfs, but they only contribute 10% of the total biovolume. Then the dwarf cells account for only minor fraction of the total energy flux of the soil bacterial communities. This is another very important. So they don't have major contribution in the energy flux of the soil, although they are genetically intact. So this is what we can extract out of here, is that they are, although they are genetically intact, but they have negligible physiological significance in the soil microbial communities, and there is a inverse relationship between cell size and cell turnover. So if we summarize all what we have discussed till now, is that there is a huge population of bacteria in the soil, which are small in size, and we call them dwarf. Each dwarf community is basically not culturable. The culturable bacteria, they account for practically all the growth and physiology of the soil communities. The non-culturable bacteria are of minimal metabolic importance in soil, although they are in big numbers. The DNA information of non-culturable communities is overwhelming as the DNA information or